Good afternoon, students.
Good afternoon, sir.
Okay. So yesterday we were actually talking about the unit 9.1. We begin with 9.1 and we briefly discussed how various methods need to be worked out to study humans, correct? Because it is unlike with the other organisms where we have enough control. With humans it is difficult, and we also discussed about various reasons for that. It could be long generation time, and also we discussed how the number of progeny, so which is a very small number with respect to the humans, affects it, correct? So sample size is always small, and also bias in finding the mating partner would create the problem. So because of that, we have to work around the various possibilities that we may have with respect to utilizing the component related to availability. So first of all, we started talking about the cytogenetics part. So cell genetics, remember what methods did we discuss as part of it in cytogenetics? So in cytogenetics, we started with the components of, first of all, bar body we discussed, right? Shweta, what is bar body?
So inactivation of extra X chromosome in females.
Very good. So it's an inactivated form of extra X chromosome. So additional X chromosome gets inactivated. Maybe explaining the concept called dosage compensation. Okay, right, you're absolutely right. So how is that done? How is bar body identified? Vishal?
Sir, the chromosomes are stained, and then
So chromosome staining is used. Okay. How does it look like? How does a bar body look like?
it's a small spot on the nucleus of a cell.
Correct. You're absolutely right. So Sunil, how many bar bodies are found in case of XXY? XXY is Klinefelter's, right? How many bar bodies are found in XXY?
Sir, one.
One bar body. Exactly. Right. What about in super females, Yash? Super females?
Two bar bodies.
Right?
Two bar bodies.
Correct. Very good. Okay. So next, we talked about these. So bar bodies are useful in identifying the gender like that, it's a male or a female, or abnormalities like extra X chromosomes can be also identified using bar bodies. Correct. Next, we talked about karyotyping. This is one of the very, very important techniques or methods that are routinely used. So in detecting various problems in chromosomes. So what do we do in karyotyping? Any one of you? So what do we briefly do in karyotyping?
Sir, we collect the samples like blood or
Correct. Any sample containing chromosomes in it, basically. Okay, go ahead.
Then we add certain chemicals to stimulate the cell division.
Very good. So we add some chemicals to stimulate cell. Next.
Then we add another chemical to stop the cell growth at metaphase.
We add something to stop the cell growth in the metaphase. Okay, so we let the cells to grow, multiply for some time. Add something like colchicine to stop it at metaphase. Correct. Next.
Then we add salt and swollen the cell.
Correct. So basically prepare the cells to burst easily in a way. Correct. by adding a hypotonic solution, less salt solution. So cell will absorb more fluid. So hypotonic solution normally sends the water into the cells. Cells will swell. So this is mainly preparing towards blasting the cells or opening the cells. Right. Next.
Then we drop the cell on that plate.
Exactly. Again, this is part of the cell opening, right? So we drop it on the slide, the glass slide. We drop it on the glass slide so that it falls on it and opens up, bursts basically. So that is what happens when this bursts, Shweta. So what exactly happens when cell actually falls on a slide like that?
The like chromosomes align themselves on the plate in the decreasing order.
Do chromosomes themselves align? When you drop
No,
something like that, do you think chromosomes will themselves align? It's all
no,
right.
sir. Themselves they won't.
No. So what happens then?
So then, like, it's a very laborious work. We have to um, mark them according to the size, according to the length in decreasing order.
You're right. You're right. But what is happening when you drop it? When you drop the cell from the top like this, what happens to the cell? What happens to the chromosomes?
So cell it bursts,
That
it
cell
bursts
bursts.
off,
What happens to chromosomes?
and chromosome spreads over the
Right.
dish.
They basically they're starting to spread around. Right.
Hmm.
When you actually drop it, they spread around. They split basically. They move around in a way. They get spread. Then we do the arrangement part. For arrangement, Sunil, what do we do for arrangement? So let's say now this chromosomes are sitting on chromosome spread is seen on the plate. What do you do next?
Sir, we arrange them from longer to smaller ones.
So how do you arrange? Do you actually hold them with forceps or anything? How do you arrange? Vishal, how do you arrange it? No, this is
By taking photographs.
exactly. We click a picture of it. That's it. So we put it on under microscope. They're so tiny that we cannot handle with the hands, right? So what do we do? Instead, we actually take a picture, click a picture of it. We'll take the photograph and cut individual chromosomes, then arrange them. Correct? Arrange them from what order, Sunil? Arrange. How do we arrange them?
Sir, longer to smaller ones.
Right. So larger one will be on the top, largest the first one, and then we organize them in the decreasing order, descending order in terms of size. Correct. So what facilitates the pairing of them? What facilitates identifying the right pair? Because we missed one component here. So is the size only criteria for arranging them?
Banding patterns.
Banding.
Such training.
How is banding pattern created? Yash, how is banding pattern created as you're saying?
Gym search train, we add color to that.
So we color it, correct? We color it. Giemsa stain is routinely used stain, or there are different methods. Either same giemsa stain is used with the reverse banding where you heat the sample first, and then centromere staining is done. Okay, where you treat the external portions, that is telomeres and the arms of the chromosomes. So like that, multiple methods are done. I don't think we need to get into the details, but just be familiar that we have something like a giemsa stain is used in order to create the banding pattern. So when bands develop, it is easier for us to match and identify the pair. That's how you organize it. And then in case if there are any abnormalities, when you arrange them, it becomes obvious to you. If you properly organize them, you know if there are extra pieces of chromosomes. You know if there's extended sizes of the chromosomes. Although it is difficult in karyotyping to have that resolution because it is limited by how good is your microscope, right? And it is limited by if you make any errors in the process. It is limited by if it drops fine, but the spreading is not great. The chromosomes overlap on each other. So like that, in that sense, it is laborious. But traditionally, this has been used for identifying abnormalities in chromosome structure. Can we find the chromosome structural difference using karyotyping? Can we see the difference? Like deletion happens, small deletion happens. Can we use karyotyping for that? Vishal?
No, sir. For small deletion, it's not possible.
Small deletion, it is not possible. We can see the gross abnormalities, bigger, better visible, because resolution is limited, right? So we can definitely see number variations. Aneuploidy we can see. Instead of 2 chromosomes, we have 3 chromosomes, trisomy, monosomy, we can see it. So aneuploidy can be easily studied. Numerical aberrations can be easily studied using karyotyping. And depending on the resolution and depending on the size of the deletion, duplication, translocations, we can also study them, but we need better methods. That is where we talked about the more advanced methods. One is called FISH, fluorescent in situ hybridization, we talked about. And what happens in FISH? I think Yash was asking a question on this, right? So can you just explain FISH and tell me what is your question?
Sir, in the fish technique, we add a probe. First, we make a probe, then we add color to this, and then we add to that probe we add in the nucleotide which present in the gene. We first have to do denaturation process, and then we have to add that. Then after this complementary basis, we can find out of this.
Correct. Just to review what you just said. So what do you do? First of all, in the chromosome, you need to know what sequences you're looking for. You need to have a knowledge of that region, which can be a limitation for this technique. Beforehand, you need to know what you're looking for. So the sequence, if you know the sequence is ATGC, you will make a complementary, single-stranded DNA for that. And this complementary to the complementary DNA, you add fluorescent probe. And when you drop that probe onto the chromosome, basically in a test tube, it will go and bind to wherever that region is. Technical details are not needed. You can forget about that. Meaning you don't need the denaturation process. How does it bind? Whether it displaces. But anyway, you thought about that. So it won't remove. It won't replace completely the two strands. So what will it do? Denaturation would allow. It will open the DNA. This probe will bind in the gap. So the other strand will remain. You are not removing it. So whenever the probe work is done, it can renature. Or anyway, we don't care about the sample anymore. We destroy it or we remove it. So basically what happens if the double-stranded DNA is there, it will open up like this. Double-stranded DNA will open up. In the gap, probe will bind. So that is enough for the probe to gain access to it. That is why we use small probes. But probe is complementary to the DNA region or the target region. So what happens because of that? So we can identify specific region on the chromosome. That is why same fish method also we learned in gene mapping. Gene mapping is you want to identify the location of a specific gene on the chromosome. If you know what gene you are looking for, you make a complementary copy for that, which is called probe. You attach a fluorescent tag to that, so it's called fluorescent probe. And then you use it to identify the specific location of the gene. Like that gene location can be identified using FISH method. Similarly, chromosomal abnormalities can also be identified using FISH method. So meaning if fish actually does bind, so we can see in case if the fish which is supposed to bind to one gene, what if we actually see more signal? We normally use controls for this. Let's say in a normal condition, fish supposed to give, let's say show one region. But in case if you saw two regions where a single fish probe bound, what does that mean? Did you understand my question? Okay, if not, I'll repeat this. So my question is this: you have a chromosome, you are looking for one particular gene in the chromosome. You added a sequence matching to that. But what happened in a normal condition, you will get one signal, one fluorescent band you will get. But in another condition, you notice that there are two fluorescent bands. What does that mean?
Duplication. Duplication.
Duplication. The genetic region got duplicated. We are finding two such regions. Fish is binding to two such spots where it's supposed to bind to one spot. So duplication event can be seen with fish. In a similar manner, you have normal component, normal chromosome, you know that gene exists. You added the probe bound one band and shown. But another component, let's say that you took it from a patient who is suffering from cancer or some genetic abnormality. And you didn't find that band. What does that mean? Fish didn't bind to any sequence on the chromosome, although it's supposed to. What does that mean, Shweta? So if fish didn't bind, no signal came from the chromosome. In the normal control condition, you found one region where fish bound. The another chromosome, which is from an abnormal, from a patient, no signal, no probe bound region can be seen. What does that mean? Lacking this fish region or a signal means what?
The genes are not present, like the genes, the
Yes,
coding of genes is not there.
Gene is not present. It's supposed to be there because it's a human gene. It's supposed to be there. So what would have happened to that gene?
Mutation?
Mutation. If mutation happens, would the whole gene disappears?
No.
No, you're right. Mutation may be a possibility, but mutation is single letter change usually. So you can still have the probe binding to that. Then what could be the other reason if not mutation? Where gene is
Deletion?
lost? What is it?
Deletion.
Deletion. Very good. So from this example, as you notice, you can simply draw pictures for this. You draw control, a normal chromosome. This fluorescent probe, fish probe bound. Okay, you got a signal. Second condition, you have two such signals. You draw two lines, not one line. Each line representing the fish highlighted region or fish bound region. So what happens? Two such signals means duplication, and absence of that bound region is deletion. So can we find structural abnormalities such as deletion, duplication, translocation using fish method or not? Okay, is it clear from this analogy, from this example, that how fish method can be used in identifying, studying structural abnormalities? Even numerical abnormalities can be found, but we just talked about karyotyping is a bit limited in terms of providing this very tinier, smaller changes, but fish is very good at that. So small changes can be identified, structural abnormalities can be identified using fish. Okay.
So can you show a diagram that we can use, or maybe?
Yeah, I think I was showing a diagram in gene mapping. If you guys have seen this, let me pull that diagram. In gene mapping, I have shown you this. Let me see.
Sir, if mutation takes place, then also it will show a highlight.
If mutation takes place, what happens? Yes. So depending on the size of the sequence, if you only have few letters, one mutation can affect the binding of that. Okay. But if the sequence itself is slightly long, one mutation doesn't make a difference. It's like this. How many bases are pairing? Let's say you have a 4 base pair fragment in the fish. 3 are pairing, 1 is not pairing because of mutation. So this may not bind. But if you are using 10 nucleotide length fish probe, then it will bind without a problem. So depending on the size of it, you may or may not see the mutation. So that is how you normally design. Are you looking for a deletion or a duplication or mutation? Depending on that, you design and you define the length of it. Okay. Okay. This image doesn't show it. 1 minute, I'll show you a good image on this. Yeah. Okay, I think I'll probably show you the gene mapping image. I'm not finding something that is quick enough for you to see it. So the image I was showing you in gene mapping part in the last unit in 9.4. Let me use that image to explain this to you. So see this image. Sharing the screen with you. Yeah, this image, if you remember, we were discussing in the class. Right, this is a fish technique we talked about for the gene mapping, right? So this is what normally happens. Look at this one particular example. So fish probe is made for this gene here, another fish probe for this gene, another fish probe for this gene. Imagine a condition, you draw a chromosome structure like this, where your probe is only looking for this top gene, green color gene. Imagine another chromosome next to it where you saw not one green spot, but two green spots. You can just draw a chromosome structure, draw one line. So that line indicates where fish is bound. In this particular case, fish is bound here. Of course, this image is meant for explaining the gene mapping. So we were seeing the locations of different genes and we constructed a gene map in the end, right? Like this. So what is the order of these genes on a given chromosome? But for the same thing for showing chromosomal abnormalities, what you can do, draw chromosome, like put a centromere, draw the arm like this, and show one spot saying that this is where fish is supposed to bind. Let's say chromosome 15, Prader-Willi syndrome or Angelman syndrome we talked about in the genomic imprinting, right? In that, what do you see? Deletion in the maternal side or paternal side can be seen depending on the disorder. So what happens here? Here, let's say in a normal condition, you draw one chromosome, say normal. Normal, you show one band. So this is where this gene should bind on chromosome 15, let's say. You show another band, case 1, patient 1, let's say. Draw chromosome and show not one but two bands. Draw two lines. So sequence which is meant to bind to that chromosome, meant to bind to that gene, bound twice on that chromosome. What does that mean? The gene got duplicated. We have two such sequences found in the same chromosome. Two signals are coming. Okay? Two signals are coming. Two signals are coming. Okay. Similarly, you draw another chromosome structure where nothing is seen. Probe didn't bind. So case of deletion, case of duplication, and a normal condition. Okay.
So unlike in the other two methods, so we have this banding pattern to identify the number of chromosomes. How do they do this here? As in, how do they identify translocation?
How do they identify translocation here? Okay, so the presence of gene, position of the gene, and the location of the gene. Because on the chromosome, on this image as you see it here, you'll get approximately the overall geometry of the chromosome. First of all, you know the position of the gene, whether it is present at the tip, let's say, and also approximately the length of the chromosome can be seen here. So normally what do? They run a lot of controls for this. So you approximately have an idea about how big is the chromosome. And especially people who have expertise in this, they keep running these type of tests. So they develop an idea about how big and how small are the chromosomes. And as they repeatedly do this, they would also know, like for example, green region bound here. It's supposed to be bound here in the shorter chromosome. But instead it is found to be in another chromosome, right? So what do they have to do? One, visual observation. Second, they have to find, they have to try another alternative method to see whether the length of this chromosome is same. Maybe restriction mapping. They use restriction enzyme to cut it, see how big is the fragment. There are multiple approaches that they can take. Okay, either manual observation of the size and the position of that band or the signal, plus maybe alternative methods to look for whether it is the same chromosome and whether it is length has changed. Like cutting with the restriction enzyme will tell you whether the size of that fragment, let's say this part moved to this region, for example, just for the simplicity, this region. What do? You know where the restriction sites are present, you make a cut of this. And you will notice that when you cut this chromosome, the fragment of so and so length has to come. Let's say 1000 base pair length has to come. But instead of 1000, if you saw 1250, 1500, you know that this chromosome length is increased. Like that, combined with another method, you may be able to find out the exact location of that. So, but you have to have a method. You have to plan something like that, which gives the confirmation about what you are seeing in this image. And normally they use not one, but multiple probes one for standard reference. So one probe always binds to chromosome 15, let's say. And if they see duplication along with the, let's say, red color probe, you also see green color probe, indicating that this additional gene component can be seen. Okay, right. But you don't have to get into the technicalities of this, how they do. But a logical manner, if at all you want to know something like this, you can definitely discuss. But is that clear? Do you understand how? And what do you think about karyotyping versus fish? Which is better? Which is more accurate?
Fish.
Which is more accurate? Right? Karyotyping can be less expensive and can help us find out the numerical abnormalities. Fish is more specific. It can show us the structural abnormalities as well with more accuracy. Okay. So we discussed also spectral karyotyping. Did you read about spectral karyotyping? So what is the difference between fish and spectral karyotyping?
Spectral karyotype is whole chromosome is of one color.
Whole chromosome is painted or colored. Very good. Fish? Only regions, as we discussed just now. Only few regions here and there. So difference is sky or spectral karyotyping is multicolor fish method. So use multi-probe or multicolor fish method can be called sky. So that you want to paint the entire chromosome. You paint the entire chromosome, like all different probes with green color for chromosome 1. You will get the whole green, chromosome 1 colored green. All probes, multiple probes with red color, you will get the red color for chromosome 2, let's say. Like that, you can color the chromosomes and identify the chromosome based on the color pattern. So sky is the, in a way, extended version of fish, where you color everything and see the chromosomes like that, where easily, if you see the color exchange between red color chromosome 2 and green color chromosome 1, translocation can be easily seen with sky, but more expensive. We need a lot of probes, and mixing lot of probes and designing that experiment is more expensive. And for that, we don't have to manually do. Computer programs can be used. This is like more advanced strategies that we use in cytogenetics. Okay, right. In the handout that I have sent to you, I've shared with you yesterday, I've also given an example for fish. Karyotyping, we have taken enough examples in chromosomal abnormalities, right? Triple X syndrome and Downs, for all those things I've shown you karyotypes. So let's talk about the example. So for the fluorescent in situ hybridization, I have briefly mentioned this. So this can be a very good and very useful diagnostic test. Fish was used to show one acute myelogenous leukemia. There is a particular cancerous condition called AML, where chromosome 16 shows inversion. So this you can write as an example for inversions that we were talking about. So AML, acute myelogenous leukemia, or you simply write a form of leukemia called AML, happens because of inversion in chromosome 16. And fish method has shown the inversion. So normal chromosome versus affected chromosome, you can see how the label is changed, how the probe region is changed, position is changed. Okay, using fluorescent probe, they have detected this. So chromosome 16 inversion that leads to AML was identified using fish. Okay, this is one. Or if you want to write something you are more comfortable with, chromosome 15 deletions that we talked for genomic imprinting, Angelman and Prader-Willi syndromes. Chromosome 15 deletions, I was just mentioning to you as an example. Okay, in the genetic imprinting. That deletion is also found through fish method. So whatever I've explained to you to draw the image, this description is given here, so that we can identify genes with increased copy number, multiple genes, meaning multiplication event, or gene loss, that is deletion event, are shown by more or fewer than 2 fluorescent dots, depending on the fluorescent dots or signals. One can identify that. Okay, right. So spectral karyotyping also have shown. Translocations can be easily studied using spectral translocations. Many cancers examples I have given you in translocations. Okay, Those examples you can write here. That sky is of great use in studying translocations. Because of the color patterns, you can easily observe this. Okay. Excuse me. The last one is comparative genomic hybridization. And again, I'm telling you, you don't have to write all of them. Karyotyping is the most important one. If you want, write fish and mention sky. If at all they ask about karyotyping. Okay, another example that I have given in the handout is comparative genomic hybridization. I'll explain the concept briefly. If you're comfortable, use it as an example method. Otherwise, it's up to you. So comparative genomic hybridization, bit complex than what we learned so far. So far in the fish, what we have done, we only put one probe, okay, or multiple probes and one chromosome. So basically one cell. But here, what do? They're comparing the hybridization means pairing. They're comparing the hybridization process. So for this, let's say that you want to identify whether a cancerous patient have a problem in his genome. So you take that person's DNA. So you collect his chromosomes, cells and chromosomes. And what do? So here you prepare sample taken from a person or a person showing the tumor or cancerous tissue and a normal person. You take normal person's DNA and also cancerous patient DNA and mix both. Okay, but what do for a normal person and a cancerous person? You take two different colored probes. So let's say green and red probes you take. So you are taking probes that bind differently to the normal person's chromosome, abnormal person's chromosome. So two probes have to be made with normal sequence and abnormal sequence. But what do here? You mix the samples and then add the probes together. So how do you interpret this? When you have two cells, chromosomes carrying normal and abnormal versions, and probes, some bind to abnormal version, one bind to normal version. What happens here? So here you notice that the procedure, as I was explaining here, so the hybridization method is used to identify gain or losses of specific chromosomal regions. It is based on simultaneous hybridization process. Both simultaneously binding, simultaneously fluorescent probes are binding to the regions of DNA. Okay, labeled tumor DNA. This is from patient suffering from the tumor or cancer, and another one for the normal reference DNA, normal DNA. So what happens here? You take the ratios, the ratio of the fluorescent staining between the tumor and normal samples. So you take the ratio of probe binding to the tumor part, probe binding to the normal DNA part. If you have equal ratio, what does it mean? Normal amount of tumor DNA in a given chromosomal region. So what do you notice here? You have, like, if it is equal ratio, both equal amount of green, equal amount of red. There are machines which can quantify it. Then what does it mean? There's equal number of tumor DNA as well as normal DNA. But if there is a decrease in intensity of tumor specific probe, let's say tumor specific probe is a red color one. Let's say you have less of red color, more of green color. What does that mean? Excuse me. Less of red color, more of green color. Sorry, less of red and more of green. Correct. Decrease intensity of the probe that's supposed to bind to the cancerous tissue, cancerous part of the chromosome or abnormal part of the chromosome. What does that mean? So in a given mixture, you have
some genes are not available, or some
deletion has happened. Gene loss has happened in the chromosomal part. So chromosomal deletion in the tumor part can be seen. In case there is an increase in the intensity of this region, duplication event has happened. So concept is same of what we talked about, but here it is done differently. Here it is done in competition with each other. That is why you have to take the ratios. So you mix the normal DNA, abnormal DNA or DNA from the cancer patient in this case, add probes that specifically bind and calculate the ratio. Higher the ratio for the normal one, then, so as we discussed here, higher the ratio for the abnormal one indicate the duplication event, lower ratio for the abnormal indicate the gene loss or genetic deletion. For that, why are you using normal chromosome? It is a reference point. It is just for the sake of comparison. That is what's called comparative genomic hybridization. Hybridization means pairing or probe binding to the target DNA is hybridization. So this is a comparative method, is called comparative genome hybridization. Okay, so this is a kind of a supplemental method, an additional method that we use here, and it is very sensitive because this ratio, small difference in the ratio can also be identified. Small difference in the ratio can be identified. Things as small as 1 MB, 1 megabases can also be identified here, meaning 1000 nucleotides can be identified. Okay, and it's up to you if you want to talk about CGH, otherwise karyotyping, spectral karyotyping, FISH are sufficient along with the bar body for the cytogenetic methods. Are you seeing the now link between how these methods are used in studying the problems or abnormalities in humans? Chromosomal problems mainly that we focused on here. Okay, right. So you have to briefly explain this. You don't even need so much to write in the exam. With this idea, you summarize, write a summarized version of karyotype. Briefly, what is karyotyping? Now, what is the principle? How is it done? Maybe in paragraph, 3-4 lines, you can write it, or with flowcharts, as you explained today to me, write the steps like that, and show a rough diagram of karyogram or ideogram or the picture where chromosomes are arranged, and show trisomic condition. Okay, and if they're asking to discuss critically, then talk about limitations of karyotyping. Put some emphasis on limitations or problems with karyotyping, the laborious nature and resolution issue, and not being able to see, fine or small abnormalities like structural abnormalities. Talk about that. Good. Okay, and where possible, you can also write. I've shown a case study here. So this is about what they're doing in Denmark with respect to Down syndrome. In Denmark, where the government in 2004 issued some guidelines where they wanted every pregnant mother should go through prenatal screening for Down syndrome, irrespective of whether she is of age 35 or even earlier than that. Initially, they said only the females at the age of 35 and above were screened for this if they are pregnant. Okay, but later what they have done, let's actually start with the, doesn't matter whatever age they are pregnant, because there may be other factors here, although maternal age has a positive correlation. With increased maternal age, increased Down syndrome we talked about, right? Keeping that in mind, plus sometimes it may happen because of other factors, not just the maternal age. So they decided to check every pregnant woman, irrespective of her age, for Down syndrome, presence of Down syndrome. They can use probe here, FISH probe, for 21st chromosome. If you get 3 signals on different chromosomes, then they know that there are 3 chromosomes, trisomic condition, as opposed to only 2 signals they should get. Or simple karyotyping, where trisomic condition because of the arrangement of the chromosomes can be seen. So Denmark government, which is actually, they have a very good public health policy, so it's free. It's free of cost for them to get such a type of, all health is free for them. It's like our Aadhaar card. You get an Aadhaar card. Based on something like Aadhaar card, you go to any doctor, they recognize you with the Aadhaar card, and government pays for your health checkup and all these tests. So government ensures that to have reduced disease burden, they came up with this idea. Okay, so that is what you notice here. What they have done, research tracked. So what the progress when they came up with this type of screening in Denmark using cytogenetic testing, what they have found out, they could track number of trisomy 21 births and use diagnostic testing, either using CVS or amniocentesis here. So the impact of the guidelines show that infants born with trisomy 21 has been halved, meaning 50% of them were only now are suffering from Down syndrome. Why? They could probably would have gone for the decision not to have the baby, identified the problem earlier. So now babies who are born with the Down syndrome had been decreased because they took the care about that already by knowing it. So the number of diagnosed before birth increased by 30%. The number diagnosed before birth increased by 30. So before children were born, they could identify them, and the number of such outcomes have increased by 30%. So already parents got to know that their children can have, and 30% of the increase have happened in the numbers. That is the benefit of genetic screening, and as genetic counselors recommend. And the number of invasive prenatal diagnostic tests done each year has decreased by 50%. So they didn't even have to go for these type of methods like CVS amniocentesis. They came up with the alternative methods. I was talking to you briefly about the like PCR methods. There are specific methods where invasive methods can be avoided. Like that, Denmark, although very small country, because of such a medical system, they are checking, they are keeping track of problems that causing big burden. So population burden this is. For a country like a small country, they have more issue there. So they're actually using the technological help to screen for the mothers carrying the babies with Downs, reducing the burden by eliminating them when they don't want or preparing better preparation towards them. And it's a country where many of the people with certain genetic problems are well taken care of. I happen to do one of my masters in Denmark, where I have seen the country where they had a great amount of health system. Okay, and they have such technology is also great. They have 4 universities which happen to be top 100 universities. Of them, 3 universities are top 100 in the world. Okay, right. So you can write something like this as a case study, or you pick up anything. It doesn't matter. I don't know whether we have in such a program in India. America should have a good program for this. You can pick up any such example. I remember taking it from one of the research articles. Okay, right. That's about the cytogenetic methods. Okay. Any questions on this part before we move on to the next method, next category of methods? The next one should keep you a bit more engaged. Okay, something you already have a bit of knowledge of.
Sir, can this trisomy, this Denmark case, since the population is small, can this be taken as a bottleneck effect?
No, not necessarily, because bottleneck effect where it must be something like a very small population where an incident have happened or accident have happened, killing killing some individuals, creating loss to the genetic diversity. So that is not the case, right? Although Denmark is small, it allows people marriages happen. We have so many Indians there, and we have people from Pakistan, so people from China. So a lot of intermixing is happening there. So it cannot be a good example for bottleneck.
So, so can deletion in genes impact the memory?
Depending on what gene got deleted. So
I mean,
can be
if someone loses its memory for 15 minutes, 10 to 15 minutes, or less, like 5 to 6, then like we can say that deletion might have happened.
Why are you thinking there is a connection between memory and deletion? What makes you to think that?
Like, we have talked about the tumor. So, I was just linking because my grandfather has a brain tumor, and like he does not go through pain or anything, but just 5 to 7 minutes loss of memory.
That is mostly because of the location where the tumor is formed. Because in the brain, we have multiple regions where memory gets stored, information gets stored. Okay, so there must be the location of the tumor is where some amount of information is probably stored. So because of the maybe physical presence of the tumor is affecting his memory ability. So it may be because of deletion or may not be. Just looking for a deletion may not help you. Because now I think instead of finding the cause, I think how to deal with tumor is the biggest thing. But the problem with tumors in the brain is it is very sensitive region. So considering to go for something like a surgery is very, very challenging.
Yes.
So I don't know whether are they considering doing anything like chemotherapy, radiotherapy to him? How
No,
are they?
no, sir. It's been 10 years. Like tumor has been diagnosed, and we haven't like done anything.
It must be a benign tumor, a tumor that doesn't spread.
Okay.
So I think they're calculating the risk. It's like this: if you do the surgery, what amount of risk you have, instead of keeping the, or keep the tumor as it is. Having that short-term memory loss should not cause much problem, right? If they go for surgery, he may end up having more complexities, more complications. So they go with the case by case and the age factor important. So all these tests we're talking about is done early enough in the age. So that we know if we can take proper care beforehand to prevent the problem or to manage the problem. Okay, right. Any other questions, guys? If
Sir,
not,
do we know in the chromosome karyotyping map? So, do we know that which chromosome has come from father or mother? I mean, the left one or the right one. So, is there anything like that?
you can look for good question. For some chromosomes like Y chromosome, you can do that, right? That is probably obvious to you, correct? So, but other chromosomes, would there be gender specific differences? I'm not sure. Maybe for some chromosomes it may be possible, but I'm unsure about it. So let me check that. So you're saying whether we can differentiate whether it's mother's chromosome or father's chromosome, right? I'm sure about that.
At least for X, I think we did mention for all the diseases, lack of maternal X and all.
So X chromosome, Y chromosome, for sure we can do. But for the autosomes, is it possible for autosomes? Is the question. Right. So let me see. I'm not looked into that. We differentiate autosomes from. Let me see if I can find an answer to that. I don't know. Maybe possible given the amount of advancement that is happening nowadays. It may be possible. Okay, right. So the next technique that we are going to go with today is pedigree analysis in 9.1. Next, we are moving on to pedigree. Okay, cytogenetic methods we finished. Cytogenetic, chromosomal, and karyotyping analysis we finished here. So now we are moving on to pedigree analysis. Pedigree, twin studies are the next two important ones. So pedigree, I think I was briefly mentioning to you about pedigree. So what is pedigree analysis? So pedigree refers to family tree or a visual representation of family tree with history of a trait. So meaning what do they study here? If you are learning about a particular trait, like for example, hemophilia running in the families. So you want to know how many people in that family tree or the family history have been affected with that. A person can go to a counselor telling her, I'm suffering from this so and so problem. Then he may ask him, like, how many family members? Do you know your father, your mother who has these features, your aunts, your uncles, your grandparents? So he would collect the information about him, his cousins. Okay, he would collect the information about them. So like that, when they find out the information like this, they can create what is called pedigree chart. So here they're collecting family information and writing down. How do they write? I'll show you that. But they create a family tree with this information. And this is with respect to what? With respect to certain character or a certain trait. That is why it is called a visual representation of family tree with history of studied trait. Okay, I will show you images, we'll work on examples. This will become more clear to you as we move on. So it's a representation or creating a family tree with respect to a character or certain trait is what is called pedigree. Excuse me. So in these charts of family tree, you will find histories with phenotypes. Phenotype means
Appearance.
the feature that is seen. In this case, hemophilia. Are they showing any known characteristic of hemophilia? Basically, no clotting of blood is a phenotype here. Okay, no, not having or not being able to clot the blood is a phenotype. Or albinism. You heard about albinism? People look white. They don't have melanin pigment. Okay, albinism, phenotype of them. So like that, history with the phenotypes and family relationship. You write mother and father, children, their parents. Like that, you actually connect the family trees with respect to their relationship. Like that, you create a chart which is called a pedigree. And of course, if you want to create something like that, there must be some standard rules for this. So they use symbols to represent different family members. They use symbols to represent different family members. I'll tell you what are the important symbols that you need to know and examples of diseases for you to understand how pedigrees work. But overall, pedigree is a family tree with information about a character or a phenotype while placing the family members with relationship with each other. Okay, using some standard symbols. For example, this is how a pedigree chart looks like. Okay, this is how a pedigree chart looks like. What do they mean? Have you ever looked at this pedigree chart before, any of you? Vishal, do you know this pedigrees?
No, sir, I have not looked at this type of chart.
What about others? Because I think you guys have different background, right? I don't assume that you know this pedigree charts. Am I right?
Sir, we have worked out the relationship on kinship terminology, but that included triangles and circles for males, females.
Right. Yes, I think in that series you use slightly different way of using these symbols. So first I'll define the symbols and then we look at it. This is a bit more complex picture, but I'll first show you some simple ones. Yeah, this image here. So are you able to see this image? Is it readable to you? So is the image readable to you?
Yes, sir.
Okay. I can enlarge this image. So first of all, we draw a square like this for a male. Male is represented by a square and female by a circle. And only note down the important ones because the examples that we use, we don't need all of these symbols. This is an exhaustive list. I'm only going to point out few ones and make note of that. So here, and it is not filled, it is empty circle here, correct, empty square here, meaning unaffected person is normal. A normal male is written like this. You know, no filling in this square, no filling in the circle for female. That means these males and females are normal, not affected by the disease genetic problem you are looking into or you are studying. Okay, this is also not needed. Triangle in this case refers to unknown sex. Okay, but this is not needed. We don't have any such examples we're working on. So male like this, female like this. Same thing for the affected male or affected female. What do you do? Filled circles and filled squares. You fill them, you color them completely. If it is filled, meaning it is affected. If it is square, it is a male, and if it is circle, it's a female. Okay, next comes the carriers. So someone who don't show the problem, who don't show the symptom, but they carry the mutation, right? So what do? You put a dot. So something in between to this. If they are carriers, you put a square and circle, put a dot in the center. If you put a dot, dot means they're carrier. They're not affected, but they carry the mutation. Okay. So ignore the next one, and even this one is not needed for our cases. This one is important. Deceased person. If you strike off these symbols, meaning that the person is dead, deceased from it. So in a family, if someone is died from the disease, they normally show it like this. They strike it off like this, meaning that person died from that problem. So deceased person. Next one, in some of these
died from the disease, sir, or died in general?
Died from the disease, because this is mainly for it is only of use when you know the particular disease harmed or affected. Correct? Because we study all this in the context of a disease. Okay, like someone died from Huntington's disease, we will show this and so show that person is dead at so and so age. Next is proband. This is something you study in the social part as well, right? You know, but you use this proband is the term you use there. Proband. Have you used the term proband in social part? No. So what do? You actually put an arrow. Proband is a person who sought help. Is a person who is affected and went for help, went to geneticist or genetic counselor for help. So this is a reference point. So you are digging information from him, his parents, his grandparents, his cousins, his children. You are starting to probe the information, starting with the proband. So proband is where you started your probing process, your investigation process. Okay, so he's affected. That is why you see his curve like this and showing the arrow. The last one that you may need here is a family. How do you write the family? No, marriage is like this. I think this is how you write if I'm not wrong in social part. So you put a square and a circle. You draw a line connecting them, meaning they got married. So these people, they are paired. Okay. So unaffected pair you see here. And you draw a line, attach it to that from the center. So this is indicating the children line. So these two are the couple. Okay. Joined through a solid line. And then you actually draw another line vertically. And from vertical line, you see multiple branches, meaning these are all their children. These people, how many children they have in this image? You draw vertical line and then horizontal line, connect like a branching pattern. Okay. So how many children they have here?
Three.
Three children. How many boys and how many girls?
One boy, two girls.
And the order is also in the birth order. First one, second one, third one. First child, second child, and third. First male, and then two girls. And generations. You put a Roman number for generation 1, generation 2. This also you don't need. Generation 1, 2 also you don't need. But in case if they show this in the diagrams, you know what they're writing. Generation 1, generation 2. Numbering is mother, father, and children. And again, if they get married, you draw another line. If this female gets married, draw another line, show structure and her children and their children. Similarly like this. Okay, adoption, we are not interested. Twins. Yes. I don't think even we use twins just for the sake of interest and writing it here. So twins, what do? When you show the children, you instead of showing a line like this and splitting it, you actually split it like this and join it. You join these two lines, meaning they are identical twins. If you don't join it, they are non-identical twins. So identical twins, non-identical twins can also be drawn like this. The attached line here between the branches or no attached. But still you can clearly see they are from the same pregnancy. Here they are from different pregnancy. Here from the same vertical line you show the split for non-identical, split with an attached line for the identical. Okay. The last one is consanguinity. If they marry within the family, they are related to them. So you put 2 lines like this. So instead of showing 1 line for the couple, you show 2 lines. 2 lines means they're blood related. So consanguineous marriages are not. Twins consanguinity also we don't use. Okay, the first few ones are sufficient for us. Mother, father, children, deceased person, carriers are enough to learn this. Okay. And with respect to the pedigrees, whatever literature you read, whose family do they talk about? What is a well-known and standard reference in pedigree, in study of pedigree? Any idea whose family is actually well-known, well considered for this?
Ego.
Sorry?
Ego?
No. Queen Victoria.
Oh, okay.
Queen Victoria's family. So Queen Victoria had a mutation for hemophilia. And in the image, if you see in their family history, what do you notice here? Her sons, mostly her children, grandchildren are affected. They thought it's curse. It has happened because of curse, but later they realized it is because of hemophilia, mutation for hemophilia happening in her. And it starts spreading to many countries. Many of her children, their grandchildren who moved to different countries started spreading this problem, the genetic problem. Okay. To understand the pattern, I'll come back to this story later. But the Queen Victoria's information has been used as a standard study for creating pedigrees. And it is actually a best case scenario because for kings and queens like that, you keep track of their history. But for a common man, we don't. So you will not remember what was your third generation before you. You may know till second generation, that's it, right? So this is one of the big limitations with pedigrees. You will not have enough information to create a proper structure and proper understanding of the genetics. And as you see here, you're only working with the available history of the family. So we are limited by how much information is available, how many people's data is being kept. And do they really know whether they suffered this from this problem? Do they have these features? I don't think people keep track of it unless they're looking for the problem. Okay. Okay. Now, how do you use pedigrees? How do you use pedigree charts? What do you think is the benefit of pedigree chart?
Find out if it's a recent mutation or whether it's there in the family.
Okay, you can find out whether whatever the effect is has it recently happened. Very good.
Hereditary or not.
Right. We can find it. These mutations, if they're hereditary in nature. So what do? You would see the source. Where did the mutation had begun? Correct. But even not only that, you can actually see what type of diseases they are going through. Is it possible to identify the pattern of the disease? And based on the pattern of the disease, is it possible to identify what type of disease this is? When I say what type of disease, remember we talked about the different patterns given by Mendel, Rigor Mendel, in inheritance patterns. What are the different ways our characteristics inherit?
Homozygous, dominant.
Homozygous dominant, homozygous recessive, sex chromosomal, autosomal. Is it possible to differentiate the pattern using pedigree charts? Let's say you created a pedigree chart. Is it possible for us to say, okay, this looks like autosomal dominant disease. This looks like sex chromosomal or X-linked dominant disease. Is it possible to say like that? Let's see. That is what we're trying to answer here. Okay. Right. So now I'm going to go through different categories of diseases. I'm going to give you features of them. Your whole goal is to see how does the autosomal dominant disease looks like? What type of pattern does it show? Do you see a specific pattern for a given type of disease based on its inheritance pattern? First, we're starting with autosomal dominant. And the basics we already covered. Imagine That is why I started with the 9.2 first, but not 9.1. Without defining what is dominant allele, recessive allele, studying 9.1 wouldn't have been easier for me, right? But we already covered 9.2 where we learned about inheritance pattern. We have done some process, so we have an idea. Now we are more familiar about these terms. So 9.1 should become easy. That is why I placed 9.1 after those units. So autosomal dominant disease. What does it mean? What does it mean by an autosomal dominant disease? I'll share all these charts with you, okay? Focus on understanding it and see if you can figure out yourself while I'm explaining this. So you draw and make your own points. Otherwise, we have an excellent textbook chapter with me for this part in twin studies. I'm going to share that document today. Okay. So autosomal dominant disease means what, Sunil?
Sir, diseases, sorry, not diseases, any trait which has not come from sex chromosomes.
Okay, so meaning it has come from an autosome, one of the 22 pairs of autosomes. Correct? Okay, right. Dominant means, what does dominant mean, Shweta?
So trait which expresses itself
Itself in what condition?
in homozygous as well as heterozygous condition.
Correct. So even in heterozygous condition, it expresses meaning, and all these things we are talking about diseases, okay? Because we are more interested in learning about diseases for pedigrees. Normal characteristics you may not need to use pedigrees, but here for disease conditions we use. So when we say it expresses in only like even in heterozygous condition, meaning even one defective, one mutation carrying allele is enough. Even though there is another normal allele along with the mutation allele, mutant allele, it is enough to cause the problem. And this allele happens to be the one related to autosomes, one of the 22 chromosomes. In that case, it is called autosomal dominant. So meaning we need how many chromosomes to cause the problem here? Two defective or one defect?
One defective.
One defective. Heterozygous condition also it expresses one defect, one carrying mutation is enough. Let's see. So look at, now you are getting familiarized with the images, correct? I'm assuming that you probably are understanding the images. So here, answer these questions. Do you see only females being affected or also males affected? So affected, what do you look for? Filled circles and filled squares you look for. Do you only see filled circles or you also see filled squares?
Both.
Both.
You see both, meaning both males and females are affected. So equal frequency of males and females. Okay. So you rule out here, it may not be X-linked or sex-linked problem. So from this that equal frequency, you come to not a conclusion yet, but it looks to you this might be an autosomal disease. We don't know whether it is a dominant or recessive, but definitely looks like autosomal because it is appearing in both the genders. Males and females are equally. Okay, right. Next, what do you see here? This is generation after generation. So this is the parent. Who is affected here? Mother or father in this pair?
Mother.
Mother is affected. So affected mother. I'll give you an example. We have something called achondroplasia. I'm going to explain you. Achondroplasia or dwarfism. So this mother is dwarf, something like this. So you see this is a picture from a family from Hyderabad. Okay. So all these family members, majority of the family members are dwarf. They're so tiny. They're extremely dwarf, as you see. Okay. So taking this as an example, you see that this person is dwarf, this mother is dwarf. And children wise, if you see how many children this couple have?
Four.
How many children?
Four.
Four.
Four children, correct? So all the branches here, as you see, 1, 2, 3, 4. Four children. And how many of them got married?
Three.
3 of them got married. Leaving out the number 3 in this generation, 3 got married. So totally 7 members, 3 couples and this. And how many of them got the problem from mother? Daughter got the problem, so son got the problem. Daughter and son equally, a daughter and a son. 50% of the kids got the problem. Okay. And then the generation after that, let's say couple one, the second generation, do they also have children with the same problem? Do they also have people who are dwarf born to them? Yeah. Unaffected people, did they inherit the problem? Because autosomes, we have two pairs, right? So here she might be heterozygous for that. She might had this problem only in one of the chromosomes. So wherever this single chromosome went, got inherited to, those children got the problem. And those who didn't receive it, like this, they didn't get the mutation in their genes. So the children also didn't get it. These children, this line is healthy. Even this child, this person, this daughter, even if she gets married, their family will also be normal with respect to this gene. But otherwise, those affected are inheriting these characters to the next generation, and irrespective of whether they are females or males. So every generation, first generation, second generation, third generation, fourth generation, problem is going on. So no skipping of generations. You'll understand why this is important, because some patterns we'll see skipping of generations. When I show you that, I'll explain that further. But for now, you can clearly see it is seen in every generation. All generations, they are showing the problem. So no skipping of generations. Second point. Third point. All affected individuals have an affected parent. Is that true? This is autosomal dominant disease. Even if you have one allele, you see the problem. Meaning those who there's nothing called a carrier concept here. Either they are affected or they are normal. That's it. Two clear, distinct, phenotypes. Either they have the problem or they have no problem. There's nothing in between. No carrier concept. So all of them have, all the affected ones, they have affected parents. They have affected parent. They have affected parent. They have affected parent. All of them. So you can see it. If there's a carrier status, that it can be seen in the next condition. Autosomal recessive condition, you can see carrier condition. But here in the dominant form, you don't. So what does it mean? No skipping generations and all affected individuals have an affected parent is indicating that, so presence of even one mutant version is enough to cause a problem. So it must be a dominant form of the character and no gender-based link here. So it must be an autosomal character. So from these numbers, all affected individuals having the affected parent and equal frequency in males and females, it is an autosomal dominant form. So by looking at this, if you get enough information like this, you can identify that this is an autosomal dominant disease. So is it possible, if you find out that it is an autosomal dominant disease, what will be the chance? Let's say someone like this came to the genetic counselor. They said, okay, in my family, many of my cousins, they say that my parents, like in this particular case, so he's the, so this daughter, for example, okay. So this daughter, this girl is the daughter of this person who is normal. But he started realizing that in their family, so many people are dwarf. He goes to a genetic counselor asking, so I'm a person, so like this, my family has this type of history. So he can ask, what is the percentage of chance that my children will have this dwarfism problem? He would ask that question. Okay, or a person who is marrying him, she decided to marry him. She can go to a counselor asking, if I marry this person, if I consider to marry this person, can my children also be dwarf? Then what would the counselor tell him if he knows that this is an autosomal dominant problem? Would he be able to tell them that whether there is a chance for them to have a baby with dwarfism or not? Yes or no? What will he say?
Yes, sir.
So what will he say? Is there a chance that having a dwarf baby for this couple?
No.
No. How could it be that?
Sir, one thing. Hello?
Yes, go ahead.
Sir, if it is a dominant trait, then why didn't four have it?
Good question. So think about it. So this is what we did in mental studies. So let's say you draw a cross. Okay, you draw a cross, right? Capital A, small a, capital A, small. Genotype for this is one capital A, one small a, one capital A, one small a, both parents. A, because we have to have diploid number.
Yes, sir, I got it.
Right. So some only if small a or a capital A is the problem here. Those who receive two small a's will not have the problem. So 50% of chance of having this problem and 50% don't get this. Okay, only when they receive the mutant allele they get the problem, and some of them are not receiving mutant allele because there is other normal allele because it was expressing in heterozygous condition. Right? Example, you can write achondroplasia. I'll briefly tell you about achondroplasia. Okay, so achondroplasia is a very common form of dwarfism. So people who are dwarf, I think 1/3 of the populations normally, or 1/3 of the dwarf people are dwarf because of this disease called achondroplasia. And achondro means absence of, chondro means cartilage, plasia means formation, bone form. Cartilage is basically the structure that we normally have related to the bone. Right? This part doesn't get formed, meaning they don't have a normal bone growth, because of which they tend to be short. Okay, and here the mutation is caused. So you can briefly write this information. Yes, go ahead. Achondroplasia, write down absence of cartilage formation. Okay, and a few key points you write down. You don't have to write down everything here. Anyway, we are studying this as an example for autosomal dominant condition. Where is a mutation? If you don't remember, that's all right. Remember it as an example for autosomal dominant condition. For this, even you can take Huntington's disease. We studied in lethal genes. You can take that as an example here. Or else, this autosomal dominant condition, and the affected chromosome is chromosome 4. So chromosome 4 mutation in a particular gene on chromosome 4. Don't even have to by heart this, but just notice that it is related to the growth. It's a growth factor receptor where growth hormone can bind to it. Particular growth signal can bind to it, not necessarily growth hormone. So mutation on chromosome 4. And most common form of dwarfism. They say that there may be 200 different types of reasons for dwarfism, and 2/3 of them, 2/3, so about 66% of them, in them dwarfism is because of this mutation. So this is a common form of dwarfism. Okay, and it occurs in both genders, doesn't matter, it doesn't affect, it's not gender specific because autosomal problem. So it affects both sexes, all races, doesn't matter, all populations in the world can get this. And interestingly, their trunk part, dorsal part is normal. Limbs are very short. They have shorter limbs, normal, dorsal part, normal trunk. That is enough. Okay, don't need the percentage ratios, don't worry. This much is enough for this example. And you also see that both males and females are equally affected. Looks like her mother is normal. She looks to be the mother here, her grandmother. And one more lady is normal here. Otherwise, most of the family members are dwarf. Look at this body clearly. You see that more or less his body size is normal, but short limbs. But to me, even normal ratio wise, also their body looks to me even dorsal area looks to me small, but that just probably they happen to be the people with the smaller bodies like that. But otherwise, definitely limbs are very short. And see, because of autosomal condition, many of their family members are affected. Okay, right. So we'll move on to the next one is autosomal recessive problem. Autosomal recessive traits. So pedigree chart for, before we talked about autosomal dominant. Here we are talking about autosomal recessive disease or recessive trait. First, I'll explain an example and then we'll come back to the chart. Okay. You can take, there are multiple examples. Sickle cell anemia we talk about. If you are more comfortable with sickle cell anemia, explain about sickle cell anemia. Or else another example here is albinism. Albinos or albinism. Albinism. I'm sure you might have seen people who are albinos around you. Have you seen people who are fair like this? No black pigmentation, no melanin pigmentation, neither in the eyes, hair, everything from top to bottom looks white. No melanin pigment is being formed. So they're unable to produce or they could not produce melanin. Melanin pigment, the dark pigment is not formed. And mutation can be because of multiple sources. It could be 9th or 11th or 15th. So a couple of regions, meaning for the pigment, we require multiple genes and mutation in any of those genes seems to be affecting the melanin production. Okay, but luckily this problem doesn't affect their livelihood. Okay, their survival is not affected, but they would suffer from some vision issues, skin issues. They are very, very sensitive to sunlight. They will have the burning sensation when they exposed to sun. Okay, so lifespan is not affected. Diagnosis can be done by appearance, visual appearance. Okay, this is a autosomal recessive condition. Either this or sickle cell anemia. We talked about sickle cell anemia, right? Shape of the RBC is affected. So hemoglobin, gene for hemoglobin carries mutation there. That's more deadly disease compared to albinism. So look at the pattern. I think you should be able to explain this to me. Now you're getting a bit more comfortable with these terms here, right? So are the parents affected? Okay, parents are not affected. So unaffected parents actually showing the process or problem in the generations. So what do you observe here? Couple of things compared to the last one where I asked you a few questions. So like genders, do you see all genders are being affected here? Are both males and females affected? Okay, what does it indicate?
Both are getting affected.
Meaning what? What does it tell you?
Autosomal.
Autosomal disease. So both the genders are equally affected. Autosomal disease. Autosomal disease because both males and females are equally affected. Next, is it skipping generations? Do you see the problem in every generation? Generation 1, no problem. Generation 2, you see a problem. Generation 3,
No problem.
no problem. Both, you see that? First cousins got married. Okay, generation 4, there is a problem. So skipping generations, meaning dominant or recessive character?
Recessive.
Why recessive?
Because only recessive expresses itself only in homozygous condition.
So to have homozygous condition, two of those chromosomes have to come together. Two of those defective mutation containing chromosomes have to come together. That may not happen in every generation. Okay, if it is coming from another person, he's also carrying the mutation. Yes, this person is affected. Right. So this person, let's say, is a carrier. So in that case, what happens? If the other person, in this case, he married, if she carries the problem, this may be because of the homozygous condition, possibility of homozygous condition. Otherwise, recessive diseases tend to skip generations, especially autosomal disease. So affected individuals can have unaffected parents. He's affected, parents are not affected. Same as here. Okay. Right. So this is autosomal recessive disorder. Examples are sickle cell anemia and albinism. Now let's see once we'll finish all of them, we'll make a comparison of sex chromosomal problems and autosomal problems. Now let's move on to the X-linked problem. Sex chromosomal problems, we'll go with the X-linked problem. We'll first starting with X-linked recessive disorder. We saw autosomal recessive. Here we are seeing X-linked recessive. Any known disease? Do you know any disease which is X-linked recessive?
No.
We begin our discussion on pedigrees today with that disease.
Hemophilia.
Royal disease, hemophilia. Hemophilia is X-linked recessive disorder. Hemophilia is X-linked recessive disorder. Gene mutation happens on X chromosome. There are multiple variants of hemophilia, but the commonly seen ones are linked to X-linked problems. Show you one more image on this. Hemophilias are of many types. Just know this, okay? Most commonly we hear about hemophilia A, hemophilia B. C also exists, but most commonly we hear about AB. AB both linked to X chromosomes. Okay, this is called a factor 8 deficiency. This is called factor 9 deficiency. So in clotting, blood clotting, we have so many factors, so many proteins participate in the blood clotting process. It's like one A converted into B, B converted into C, C converted into D. Like that, every step we need a protein factor to convert it. So like that, there are many of which some people actually show mutation in formation of factor 8 or gene coding for factor 8 is affected. If factor 8 is affected, this is called type A hemophilia. If factor 9 is affected, it's called type B hemophilia. If factor 11 is affected, it's called type C hemophilia. But remember only two, hemophilia A, B, factor 8, factor 9 is enough. Both are linked to X chromosome. Otherwise, there are also hemophilias, as you see, connected to autosomes. But well-studied, well-known examples are these two. Excuse me. Okay. Similarly, we also have color blindness. We didn't discuss, I think, color blindness yet. In genetic markers, we are going to talk about color blindness as an example. So color blindness is also a type of X-linked recessive disorder. Sorry.
Hemophilia A and B, there is no difference phenotypically.
Phenotypically, you don't have the difference. Cause is different.
Okay.
Okay. So color blindness, another example for sex-linked disorders, because if you know a couple of examples depending on the question, ensure to have at least 2 examples with you to write. Okay, color blindness. So what happens in color blindness, as the name suggests, so what happens in color blindness, some people cannot differentiate colors, especially red and green color blindness it is called. So they don't know the difference between red and green. To them it looks like gray, or it looks both as green, let's say. So they cannot see that color. I actually had a colleague like that when I was doing my PhD. I had a colleague, and when someone creates the graphs showing the research data, he could not see red and green. For them, both were same. And my supervisor was used to, my supervisor used to make fun of him. He would ask him specifically, "What color do you see here?" So because of he's also biochemistry faculty, so he knows that very well. So he knew that he's not able to see the colors. To them, it looks like gray sometimes, just gray, no color differentiation, because of a mutation on X chromosome. Right. In either of these cases, what we need to know is a pattern. So tell me, what pattern do you see here? For autosomal diseases, normally we don't use the word carrier. So we say sickle cell trait or sickle cell anemia. But for X-linked disorders, we say carriers. So what do you see here? Mother is a carrier. She's a carrier. She's not affected. She doesn't have hemophilia, but she is a carrier. And what about her children? So who is getting affected in the children? She has a girl and a boy. Who is affected here?
Boy.
Boy is affected. Next generation, how many boys and how many females are affected next generation? Third generation.
Again boy.
Again boy. Fourth generation. Again boys. So it looks like there is a curse going on in their families. All boys are affected. So what type of genetic disease is this? And why only males affected? If you really understood the concepts of Mendelian genetics, you should be able to explain this. Why only males affected? Very simple logic you need here. So it is more commonly seen in males. Affected phenotype is seen commonly in males. It doesn't mean that females are not affected, but it's more common to see that males being affected by X chromosomal or X-linked recessive disorder. Why is that? So mutation is carried on what chromosome?
X.
X chromosome. How many X chromosomes are present in males?
1.
1. And who gives that X chromosome to the male? Sons receive X chromosomes from whom?
Mother.
Mother. So mother is a carrier. She carries one X chromosome normal, one X chromosome mutant. And 50% of chance, because she can give normal X chromosome to some children, mutant X chromosome to other children. So 50% of her children can have her mutant version of X chromosome. And males only have one X chromosome. There is no other copy to compensate. There is no concept of carriers in them. There is no concept of carriers. It's called hemizygotes. They are not homo-heterozygotes. There is no concept of homozygotes, heterozygotes in males with respect to XY chromosome. Only one X, one Y. They have only one condition, one of each. This is called hemizygous condition. Hemizygous, sometimes you see in the textbooks. Hemizygous, H-E-M-I, zygous. Okay, so if this son receives one defective X chromosome from mother, he becomes hemophiliac. Only one defective X chromosome is needed in males. That is why usually males are affected with X-linked recessive disorders, and the females can be carriers. Because in this case, this mother gave one mutant X chromosome to another girl baby. She also became a carrier. Her son, because she gave the defective X chromosome to that son, he became hemophiliac. And him, so this person's grandchildren, their great-grandchildren are carriers. So whose chromosome are they carrying here? These two children, two daughters, whose chromosome? Two carrier daughters carrying whose chromosome? Mother or father?
Father. Father.
Father.
Father in this case. But normally, females carry X chromosomes from father or mother?
Mother.
All of you. So mother. Sunil, X chromosome in females come from father or mother? Shweta says mother.
Sir, it comes
One
from
from
father.
each.
Father is determining factor.
X chromosome. Who? Okay, father is determining factor, but there's no gender concept here. In here, it's a female. There are two X chromosomes. Where are these two X chromosomes coming from? Can both X chromosomes come from the same parent?
No. One from father and one from mother.
So as I rightly said, Vishal and Shweta, as you recognized, yes, two chromosomes. You'll see one from father, one from mother. Females have two X chromosomes, one coming from each parent. In this particular case, the mutant version came from the father and other normal version came from the mother. In males, males always receive Y chromosome from the father. Father is not giving his X chromosome to the sons. So sons are not getting affected here. As you see it, sons, his son is not affected. Son, he resulted in the carrier condition. His son is not affected. Both sons are not affected because X chromosome is not gone from him. But whereas mother gave her X chromosome to the son, so son is affected. So always it goes from mother to the sons. And mother can be carrier and son is affected even with one mutant version. And when is it possible to have girls carrying the hemophilia condition? In what scenario does a girl can become hemophiliac? Meaning you will see the circle, filled circle you will see. In what condition will you see that?
Sir, mother is carrier and father is affected.
Affected father, carrier mother, or both hemophiliacs. Both mother is affected, both her copies, X chromosomes are affected, and husband also affected, or both of them. Father is affected because there's no carrier condition for the father, mother is a carrier. What percentage of children will have it? If mother is a carrier, father is hemophiliac.
3:1.
3:1. Why 3:1? So we will have only one normal X chromosome, two abnormal X chromosomes. Correct? So one version of the X chromosome would result in this. Remaining two parts of it will result in the affected. Okay, right. So do you see a specific pattern here? Is this pattern different from autosomal recessive disorder? What differences did you see between autosomal recessive and sex chromosome or X-linked recessive disorder?
The X-linked recessive is skipping generation, and in this, males are getting affected.
So whereas in autosomal problem, there was no difference, correct? So this is autosomal. See, roughly equal amounts of males and females are affected. Here, more commonly males are affected, and here generations are skipped. Okay, it is also skipping generations. So what is common to that and this? Why are they both skipping generations? What is it indicating? Skipping generations indicate what?
The recessive.
Correct, recessive disorder. If it is X-linked, males are affected. So gender-based in a way, and skipping generations. Sign of X-linked recessive disorder. Okay, good. Logic, usually you will find the logic. Only the problem is you will forget it. So you have to revise this content briefly. Okay, I'll send you after the class the handout for this. Read it. Last condition. No, not last. Of course, we have two conditions. X-linked dominant problem. First question. Is the gender basis here? Are only females affected or only males affected here? So you see both females and males are affected. Okay. Is it skipping generations? Is it found in all generations or is it skipping generations? Found in all generations. So dominant or recessive character then?
Dominant.
Dominant. Seen in both males and females, no skipping of generations, and more females are affected. More females are affected. Why? Because females receives their X chromosome from either of the parent. Father can give that X chromosome, mother can give that X chromosome. So in this particular X-linked dominant case, you see more and more females being affected. Unlike the previous case, X-linked recessive, more males are affected. If more females are affected, X-linked dominant. More males are affected, X-linked recessive. This is a key thing that you have to remember. The last point here. Affected male will have 100% affected daughters. So you see here, this particular scenario, father has his disease. I'll show you a couple of diseases for this. Father has a disease. And what do you see? All his daughters, all their daughters have the problem. Because X chromosome has to go from the father, and father has the effect, he's affected. So he has mutant version of the X chromosome. All children have received, and although they received a normal X chromosome from the mother, because in heterozygous condition, the dominant character gets expressed. So it doesn't matter whether mother gave the normal copy or not, because father gave the mutant version, all of them are in heterozygous condition for this mutation, and all of them show the problem. So affected males will have all 100% daughters affected. So is it enough points to detect whether it's a dominant character or not? But remember this, how many gender-based effect is there? Is there skipping of generations are seen? And within the X chromosome, are only males affected or only females affected? About 2 to 3 points for each of them, if you can remember and know the logic, you can derive all pedigrees for all of them with no problem. But in the exam, so far I have seen that they ask combinedly. So talk about karyotyping, talk about pedigree. Just briefly give an example of pedigree chart. So dominant disease, recessive disorder like this differentiates. Or autosomal dominant, sex chromosomal dominant difference like this. Like that you write. Okay, comparative analysis, 2 images like this. You don't have to draw so many of them. Draw 3 generations approximately, show the problem, that's it. And explain it with 3 points like this. Okay. Any examples of X-linked dominant characteristics? Very few are there, because normally dominant problems are less common. Why? Because dominant will affect more people. So many of them don't survive. So dominant X-linked or autosomal dominant characteristics are less common. Recessive disorders are more common in populations. Because recessive, at least one normal copy will be there to compensate for the abnormal copy. So the survival may not be affected in some cases, majority of the cases. So dominant diseases are less common, recessive diseases are more common because we can tolerate. Okay, know this. Many examples, but none of them are very, very familiar or simple for you to remember. There is something called vitamin D resistant rickets. Have you heard the rickets problem? Rickets, nutritional deficiency disease. So weak bones, people with vitamin D deficiency, so they have weak bones or fragile bones, they easily break. This is called rickets. And vitamin D resistant rickets. So it is not depending on vitamin D. It's not because they are not receiving vitamin D, but because of genetic abnormality. It is called hypophosphatemia. Hypophosphatemia. Okay. Hypophosphatemia, vitamin D resistant rickets. Or fragile X syndrome we talked about. Fragile site mental retardation. So this is also an example for X-linked dominant character. Duplication event that we discussed. X chromosome. Forget about other diseases, you cannot remember them easily. So this is enough. Either hypophosphatemia or fragile X syndrome leading to mental retardation. Okay, right. Y-linked. So pattern we are learning. So it is easy, much easier. Who is affected if there's a Y-linked disease? Must be obvious. Right? Only males are affected. Y chromosome is only present in males. So all males are affected. But how do you differentiate it from X-linked recessive disorder? In the X-linked recessive disorder, the problem goes from mother to the son. But here, males, it goes from male to male. So males will have the problem, father will have, child will have. This father will have and child will have. So always you see that the problem is going from father to the son, father to the son. And no concept of carriers. And no concept of skipping generations. So everything, only males and all males, affected males have affected fathers, no skipping generations, meaning Y-linked. Very, very rare phenomena. I'll tell you why, but this is what it is. Okay. Next. Any diseases for Y-linked cases? Sorry for writing, I can wait.
If like affected male don't have sons, then
So what do you think will happen?
generation could be skipped now.
Okay, skipping generations refers to having children who is not affected. Not having children is not qualified to be called skipping generations. There must be a generation to check, right? And for sure, males. They have to have sons. If no sons, we don't know whether it skips a generation. You say unknown. We don't know whether it skips a generation because females are not affected, but males are not there. So we don't know. And the problem will stop there in that family. Okay, right. Any example? I think Y. So Y chromosome is very short. If you have seen from the karyotype, Y is a very, very short chromosome. It's a very small amount of, they say 50 million bases, still very, very small. One of the smallest chromosomes that we have compared to the X chromosome especially. So very few genes are there, very, very few genes. So few genes means few mutations that are possible in them. So Y-linked diseases are very, very rare. Y-linked diseases are very rare because it's very small and does not contain many genes. So diseases are very rare. We don't have, I think we don't have diseases that I could come across, but few characteristics are there, few traits are there. One well-known trait I'll explain you. Okay. So this is hypertrichosis pinnae. Hypertrichosis meaning having hair. Sometimes you see people having hair on the ear pinnae. This is called pinnae region. This region is called pinnae, the outer opening. Okay, on that some males actually show hair. You never see this in females. You may see mustache in females, you may see baldness in females, but you never see this hair appearance in them because it's Y-linked character. And on this hair pinnae, you notice that there is hair that is grown. Have you seen that? Have you seen this character? Some males actually have this is Y-linked character. So their children will also have this character. Sons, not females, sons. Okay, this is a known example for Y. So with that, we finished talking about pedigree charts for all possibilities here. So now, applications, uses of pedigree charts. So based on the applications we discussed, can you tell me how useful is pedigree charts and where is it useful? I think I asked this question in the beginning. Now you have the examples that you went through. Tell me where are these useful? What is the use of pedigree charts?
Genetic counseling. In genetic counseling.
Part of genetic counseling, disease pattern, genetic disease inheritance pattern can be derived from pedigree charts. And by knowing the disease pedigree charts, one can also identify the risk factors running in the families if they have a children. Because once you know the disease pattern, you can calculate what percentage of children can have that problem. If it is a chromosomal dominant or Y chromosome or autosomal dominant and recessive. So this, okay, it is very, very useful to trace genetic disorders, to identify the problem and trace the genetic disorders in the families. They're easy to build. Here you only have no invasive procedures are needed, right? Just have to collect the data. Easy scientific analysis of inheritance of genetic characteristics. Okay, so to trace and identify genetic problems is and a carrier screening. You can also identify the carrier analysis can be done. So affected people, carrier people can be calculated here. because of not relevance here, but in animals, they can also use this to perform breeding. Select the right species, a desirable species for breeding. So family history can be taken for choosing the right mate for the breeding process in animals. But for humans, basically this is mainly for, they can probably use it for as a method for qualifying or disqualifying someone for marriage, right? What is the percentage of chance having the disease? So do you think this is useful for Down syndrome and of or triple X syndrome? Is this pedigree charts are useful for diagnosing those problems? Diagnosing chromosomal abnormalities?
Would they be diagnosed phenotypical itself?
Okay, otherwise, alternative phenotype. Is pedigree a potential source?
They if they are heritable, I think can be used.
Good question. Are they heritable?
No, I think
No,
not.
they're not heritable. They're happening because of a random event during the segregation process. They're non-heritable as far as our understanding goes. Okay, so pedigree charts are of no use for understanding or identifying chromosomal abnormalities. And what is most useful for chromosomal abnormalities then?
Karyotyping.
Karyotyping, cytogenetic studies. So the one we studied before was mainly or mostly important for the chromosomal abnormalities. This is mainly important for the genetic diseases which are caused due to mutations. Okay, so when you write the diagnostic methods, remember this logic in your head. For chromosomal abnormalities, if you write pedigrees, then that's a mistake. Okay, so you have to know this logic. The last part, problems or issues associated with this or limitations of this. So you see Donald Duck family tree here. Okay, so what do you need for accurately predicting or creating pedigree charts? Data. Do you have your family members' data with you? How many generations of information you have with you? Just to see how much we keep track of information. Do you keep track of information? Do you know your great-great-grandfather? You probably know your grandfather. You probably know great-grandfather, maybe, if your father or grandmother is there. But great-great-grandfather, we don't know. Right? We usually keep track of about 2 generations. Is that enough information? Maybe not. And second, you may remember their names, but you may not remember the diseases or problems they may have, unless if they keep relating that to the grandfather. He had this problem, that's why we got it. Or grandmother, that's why we have it. Otherwise, you may not keep track of this. That is why royal families database had become very useful tool for this, because they do keep track of all these things. They write their history, how many children, how did they die, what did they done, who did they marry to. All that information, inheritance, legacy will be kept. So, but for normal families, the biggest challenge is record keeping. So, and reliable record keeping. Knowing this is the problem and writing it exactly what it is the problem. And the other limitation. So, creating accurate chart requires enough data, so enough record keeping. Okay. And another one is disease relative. Sometimes people die, so loss of information. So people may die from the problem, so we would not know whether they suffered from this or they had some other problem. And the last example, last point is family size is small. We may have 2 children, sometimes even 1 child. If you go to China, they will only have 1 child. Not enough possibilities to come to a conclusion. Statistically, very, very difficult to come to a conclusion with 1 child. 2 children also same. 3, 4 may get better. So family size is also getting smaller and smaller. So pedigree charts of not of great use if we don't have proper information, proper number of people in the family. Okay, the conclusions will be, conclusion cannot be derived. Okay, right. This is the limitation of pedigree charts. So with that, we'll stop the class here. So today we finished talking about the cytogenetic methods, and also we discussed the pedigree charts. In tomorrow's class, we are going to focus on the twin studies. We're going to focus on how to study and when to study twins, and what type of information can twins give us. That is going to be different from what we studied so far. Cytogenetic methods and these methods will not give us that idea. Okay. Any questions, guys? Any questions on what we discussed? Are you able to follow this? The examples are good enough for you to understand these concepts. I know some of the technologies like CGS, some of those are a bit complex, but you can choose not to talk about them. Okay, but I just have to cover enough. So I was giving enough examples as per the literature, but otherwise these examples should help you understand this and draw the clear conclusions. So you should be able to summarize this information. Okay, I'm going to share this document as well as this document as well as I think I should have one document with the information on this, plus one reference chapter for you to read. It's a textbook, genetics textbook chapter, not too complex. Okay, it gives nice case studies, examples, especially for twin studies, karyotyping. This, all these things can be studied from there. If you have time, if you want to read more, especially for the next class, twin studies, you can read from that. Okay, I'm going to post these two documents. Okay, so we'll have a class tomorrow. So I'll announce. Normally, if I'm delaying by 15 minutes or so, I will let you know beforehand. But otherwise, around 3 to 3:15, we usually have the class. If any change, any big change to that, I will let you know beforehand. But tomorrow I'm planning to take class. Okay, so Sunday we will have the test. Correct?
Sir, just to get an idea, so how many? By when do you think we'll get done with the classes? Since we want to plan for the rest
Right.
of the.
Yeah. So to answer that question, so basically we are finishing 9.1. Right, we finished 9.1 to 9.4. 4 units are done, 1.7 done, 1.4 is done. So for race and racism and markers, ecology, epidemiological anthropology, we require about 3 classes together. So 3 and here 2, 5 classes, 7 classes. Here for sure 3. So 10, 13, 2, 16, and then yeah, about 18 sessions we need. So about 3 weeks to 4 weeks time we need. Okay, so let's say 4 weeks to be on the safe side. So we'll finish approximately by February 20th. So I'll aim to finish by February 15th, but about 3 to 4 days here and there. Okay, that is the timeline we are looking for. So we have to finish everything. Okay, how are you doing on social part? Is social part done? Is it finished?
We are at tribal paper 2.
Okay, how many classes you need as per where you are? Is it known?
No, sir. No clarity on that.
Will take so much of time on this. So yes, I think are you good with the test part? Right, I'll mostly focus on the questions that I might have mentioned to you are the past questions. Go through the past questions, so you will know what questions I'm going to ask in a way. Right, prepare for that. In this test, I'm mostly looking onto your way of presenting information, your writing style. Okay, writing style and how well you understood the concepts is what I'm going to focus on. The first test. Correct, right, guys. Thank you.
Thank you, sir.
Welcome.